Hi everyone, I'm Hassan from the Bo Wang Lab, and I'll be guiding you through a short tutorial on the COVID-19 genotyping tool, or CGT. We developed CGT to really summarize and distill key information from whole genome sequences that have become publicly available since the onset of the COVID-19 pandemic. Since the onset of the pandemic, research sharing has been a great initiative within the community, and this has resulted in over 10,000 publicly available whole genome sequences of SARS-CoV-2 viral isolates. And so what we aim to do is to distill information from these sequences in 3 succinct visualizations, which I'll go over. So what you see here is the user interface for CGT. And it consists of a main panel, which includes the interactive visualizations, including UMAP, MST, and SNP, which I'll go over in a bit more detail in a second, and the side panel, which has an input panel where users can actually upload their own in-house whole genome sequencing information for SARS-CoV-2 viral isolates and view that concurrently with the publicly available whole genome sequencing data through the 3 visualizations that we have. Within the input panel, users can also toggle metadata to be overlaid on these visualizations, including region of sample origin, country of sample origin, and collection date of the sample. The details panel summarizes the analysis pipeline for CGT. And it also has some useful links for users that want to see further documentation, primarily through the GitHub. The data panel goes over the data sources for CGT. As I've stated, our whole genome sequencing data is taken from the GISAID platform. We use metadata from the NextStrain NCOV repository. And we also have a statement on user privacy, indicating that none of the sequence information that users upload is actually stored or cached in any way. We simply process the data and update the visualizations that we have. And so now I'll go over the 3 visualizations. So as I've stated, the visualizations are completely interactive. We can do things like zoom in on certain regions and clusters, and zoom back out using the autoscale function. We can subset certain regions by double-clicking them, such as South America in this example, and we can double-click them back to bring back all of the data. And so this is done through the Plotly interface, and it's a great way to really get a more fine-grained visualization of the data that's being presented. And that's especially important because there are so many whole genome sequences being analyzed through this platform right now. So UMAP is short for Uniform Manifold Approximation and Projection. And what UMAP can really be seen as is a way to visualize DNA distance between whole genome sequences in a 2-dimensional space. So it projects the high-dimensional sequence data onto this 2-dimensional manifold. And each point here is one whole genome sequence that's been uploaded to GISAID. And so the distance between these points can be really thought of as the genomic distance between the viral isolates. And so, for example, if we want to interact with the UMAP visualization and see some more fine-grained information, we can zoom in on this cluster. And once we do that, we see that this cluster of sequences primarily comprises of North American sequences, some sequences from Oceania, and 1 sequence from Europe. If we want to see more fine-grained information on region, we can change the metadata to country, and the visualization will refresh, and then we can zoom back in on that cluster we were looking at previously. And we can see that in terms of the North American regions, most are from the US, while some are from Canada. The 1 European sequence was from the United Kingdom, and so on and so forth. Now let's move on to the MST visualization. MST is short for Minimum Spanning Tree. And so for this visualization, what we do is we generate a network of the sequences, once again using DNA distance, similar to like we did in the UMAP. And we take the minimum spanning tree of this network, which can be thought of as the shortest path we can take while also connecting all of the nodes. The nodes here correspond to the viral isolates, the whole genome sequences themselves, and the path corresponds to DNA distance. And so in epidemiological studies, minimum spanning trees have been used to determine the most probabilistic transmission pathways in epidemiological networks. And so we thought that this would be a very apt visualization to summarize SARS-CoV-2 viral isolates. And so with the MST, we see that there's a very large, very dense network because there's a lot of whole genome sequences available right now on CGT. And so what we can do is we can zoom in. And once we zoom in, we'll see that there are many hubs within this network. And for example, we see that there's one hub here where it's the central node is a sequence from the United Kingdom, and all of the nodes sort of connected to this central node are sequences from Iceland. And so within the MST, what you'll typically see is very large hubs, some smaller hubs, and then also some very long chains of transmission. And so this can be used to infer things like transmission pathways. Hubs can be thought of as corresponding to outbreak epicenters, although this information is largely dependent on the accuracy of the sequencing and multiple alignment, so it should not be taken at face value that, in fact, this individual, which the viral isolate from the United Kingdom originated from, spread the virus to the individuals in Iceland. It should not be taken at face value, but it can be thought of as being correlated to that information. The last panel we have is the SNP panel. In this panel, what we do is we summarize the most frequent single nucleotide polymorphisms within the structural protein coding gene regions. And so the structural protein coding genes correspond to the envelope, membrane, nucleocaspid, and the spike protein. And so we really aim to look at non-synonymous mutations, which are mutations that cause changes in amino acid sequence, which can potentially affect the protein structure itself, leading to phenotypic changes. And we track these because changes within these structural regions are especially important for vaccine development and attenuation of host immune response. And so similar to the other visualizations, we can toggle any type of metadata that we like. And we can see that for each of the top variants, what we do is we present the top 9 most frequent non-synonymous variants within these structural protein coding gene regions. We can observe the allele frequencies. And so, for example, if we look at Africa, we can see that 98% of the sequences that originated in Africa are of the C nucleotide at this position within the nucleocaspid protein coding gene region. The T corresponds to a missense variant, so we have a C to T substitution. And we see that for Oceania in particular, there's a very high minor allele frequency for this variant. And so now I'll take you through the uploading of user data. So as an example, I'm going to upload one sequence in one FASTA file to indicate to you how these visualizations are going to change. Processing for this data can take up to 15 minutes, but what I can do is I'll go ahead and upload the data, and we can see that the upload is complete and these visualizations are processing. And I'm going to go ahead and skip 15 minutes ahead. Okay, so our visualizations have finished processing. As we can see, the colors for our UMAP visualization have changed, and we've incorporated our uploaded sequence as novel 1. And so this is how the nomenclature works for user uploaded sequences. For each of the up to 10 sequences users can upload at a time, it'll be named as novel followed by the number of 1 to 10 for that given uploaded sequence. And so we can see if we double click on novel 1, we see our sequence falls within the initial large cluster observed within the GISAID data. Similarly, for a minimum spanning tree, we can see where our sequence falls. It's within this area of the network. For the SNP tab, we can determine corresponding to the top 9 most frequent non-synonymous variants within structural protein coding genes, what allele does our uploaded sequence have, or what alleles does it have. And so we can see for all of these variants, our sequence does not have the allele corresponding to the minor frequency within the population of SARS-CoV-2 genomes. And so this is how users can upload their data and see where it falls with respect to the public data. And that sort of basically covers the basics of the CGT tool. We're always looking for feedback, and so we'd love to hear what your opinion is on this application. If you have any specific recommendations or problems, you can open up an issue on the GitHub. If you have any general feedback, you can forward that to the email within the details tab. And so we'd like to thank all of the researchers on GISAID that uploaded their data, their genome sequence data, and made it available for us to use. We'd also like to thank the NextStrain team for making their parsed GISAID metadata publicly available so that we can use it. And that'll be all, and we hope you enjoy using the CGT application.